For this video, we're going to focus on the basics of mitosis. The previous videos we watched are great. I think they do animations better than I could ever do, and the level of detail they go into is great. The Amoeba Sisters do really great videos, but I want to show you the details that I'm expecting you to know for my class. So this is going to be a little bit more vague, a little less detailed, but it's going to help you narrow down what you really should know by the end of this unit. Because you're not expected to know everything that you saw in those Amoeba Sisters videos. So with that out of the way, let's go ahead and go through it. Our mitosis basics. So on this left-hand side here, we have a chart that shows the cell cycle, and it shows both interphase here, which is everything outside of that blue wheel. So we'll go through the three phases of interphase, and then we have the mitosis phase, that M phase, ending with cytokinesis. So we're going to go just the basics of each of these steps so you're aware of what happens. You should have your template of notes set up, but make sure you write down everything that I do as we go through it. So the first thing that happens during interphase, again, the cell's not dividing here, but it is preparing to go through that mitosis phase. So the first thing is G1. That's the first phase, and this is growth. The cell grows larger. And this is really important because if cells don't grow but they keep dividing, every new cell will be smaller than the one before it, and eventually they'll be useless. So the cell must grow prior to dividing. S phase is when DNA replicates. So the DNA doubles. We have 2 copies of everything, and if you remember, chromosome number, the chromosome number doesn't change. Move this over just a second. So chromosome number doesn't change. We double the number of chromatids. So when the DNA doubles, it will essentially stay connected at a centromere. So we don't double our number of chromosomes, we just double the number of chromatids. And then when those separate, they become their own chromosomes again. And then we move on to G2, which is the cell grows even larger. So in G2, the cell grows larger again, further preparing for that mitosis. The one I didn't write down is this G0 phase. It's essentially just a resting phase. So not all cells reproduce, or sometimes cells need to rest longer before reproduction. So it's basically just a delay before G1 starts. But then there's cells like neurons that never ever reproduce, so they always stay in G0. So not every cell in our body goes through mitosis. After G2, we move into the M phase of the cell cycle, which is the mitosis phase, and it's where they actually divide. Remember, DNA is already replicated at this point, but the first phase of mitosis is prophase, and here DNA folds into chromosomes, and the nucleus breaks down. So the only way to get the DNA out of the nucleus is for the nucleus to break down, and so that there's no tangling issues, the DNA is folded into chromosomes. So that is prophase. For metaphase, the chromosomes line up in the middle of the cell, and spindle fibers attach to the chromosomes. So those spindle fibers, which is what you see here in this little drawing, those are attaching to each side of the chromosome, to each chromatid they attach to, and they attach at a point called the centromere. So you don't need to have that memorized, but they do attach to a very specific point on the chromosome, which is called a centromere. During anaphase, the chromatids, remember that's one half of the chromosome once it's been duplicated. So the chromatids are pulled apart and they move to opposite sides of the cell. So it is the spindle fibers that are pulling those chromatids apart, and they're moving towards opposite sides of the cell. In telophase, the nuclear membrane reforms around each set of chromosomes. So if you look now on each side of the cell, we have a nucleus, and the chromosomes from the original duplication are now back inside the nucleus. We have our DNA back in the nuclear membrane now. And then the very last phase is cytokinesis. And in cytokinesis, the cell actually divides into two new cells. And the terminology here, the first original cell is called the parent cell, and the two new cells are daughter cells. So that's just the terminology for this. And then after cytokinesis, those cells would go back into interphase, and they go back into the G1 portion of interphase, where they would start to grow. Or they might go into G0, where they would rest, but either way, the cell cycle continues. Hopefully this helps you out with just the basics of what you need to know for mitosis for this class. As always, thanks for watching.